So now I would like to welcome Brendan Blumenstiel from the Broad Institute.
Thanks to Twist for the invite today to come speak to you guys, and thank you to all of you for spending a few hours in a conference room, not poolside eating lunch. I like to think that I would do the same, but I'm not sure I would have. But anyway, this is a great opportunity to discuss. So we've sort of been thinking about Twist for a long time. We were sort of engaged with them over a year and a half ago to start thinking about how to leverage their technologies, specifically in the blood biopsy space. That's now evolved. The applications that we're actually thinking about have evolved quite a bit, but I want to kind of just really just do a summary of our experiences, what we're trying to do, and where we sort of see the stuff going. So just to give you a little bit of background, so a lot of you have probably heard of the Broad Institute. Specifically, the Broad Genomics platform is a group that's made up of Broad Genomics is made up of the Genomics platform, which is the research and clinical operations side and research, technology development, R&D. But then we're also, tied closely at the hip to another platform, the data sciences platform. Since we're generating such huge amounts of data, those two groups working together is what powers our ability to, impact science. A lot of people have sort of seen this, and the Broad Genomics is a huge sequencing center. We've been in the sequencing game for a long time. If you actually look at some of the numbers, I think they are interesting. We've sequenced over 122,000 whole genomes at this point, over 400,000 exomes, and we have 2 million samples, or more than 2 million samples in storage right now. And we're now at the point that we've generated over 21 petabases of data. The lion's share of that's been in genomes and exomes, but of course we do RNA-seq arrays and targeted work as well. We have a lot of NovaSeq, so we're doing our best to fill in the samples. One point to make is that statistic about the yield. I think we're now at about generating a whole genome's worth of data about every 12 minutes or less on average. The big driver for the work that we've been doing with Twist has actually been in the blood biopsy side. This is a close collaboration between the cancer program at the Broad and the genomics platform to sort of scale processes that have been developed in a smaller, sort of more academic side of the lab. A lot of the work that we're doing kind of hinges on the realization that whole genome sequencing of cell-free DNA does show high concordance with the actual metastatic tumor. And because of that, we now realize that we can profile tumors from circulating cell-free DNA in patients. Now, whole exome sequencing obviously still has a decent amount of cost, especially if you have a sample that does not have a high tumor percentage. One of the methods that we incorporate here, one of the tools, is we actually now perform an ultra-low pass whole genome sequencing of a library to determine that tumor purity. A sample that has really no tumor in it, if you're going to bring it to deep coverage in a blood biopsy sample and exome, is awfully expensive to not really get an actual result. So we found that we have like a good upfront QC for what samples then we can then justify bringing to depth in something like an exome. We'd like to think that all the samples that we collect are over like a 10% tumor purity, but in reality they're not. So the actually approach at Broad is we are actually receiving in lots and lots of blood tubes or pre-fractionated plasma. Fractionation happens in-house. Cell-free DNA library is made, and then from that library, the goal is to do that upfront low-pass, ultra-low-pass QC, and then be able to determine the fate of that library or of that sample into an actual sequencing product. Things with high tumor percentage could go to a whole exome, but things with an extremely low tumor purity, either below the level of detection or just like an unknown, we know that we need to go into a targeted panel and go extremely deep to actually be able to detect with some sensitivity those minor, those low allele fraction variants. So cell-free DNA as a whole is interesting to work with. The tight size distribution that we typically see around that 166, 170 base pair size, actually, when you want to go extremely deep, limits your ability to see unique depth because of sort of a reduced start and stop distribution of fragments. So that really does drive us into the world of using unique molecular indices in that library construction method so we can identify unique original molecules rather than duplicate molecules. That helps us sort of, one, identify unique molecules, but also it helps with the elimination, or at least error correction, of PCR or sequencing errors. And overall, just increases the accuracy. The image, the IGV image of these reads isn't that great on the screen, but without building a consensus read, you do have a lot of PCR or sequencing errors. Using consensus read construction, using UMIs, you end up with an extremely clean data set. So ultimately, our aim has been to develop a single, and this is the key to what we're doing, a single workflow. Since we have such high scale, what we wanted to build is a single workflow that can support a wide sort of range of different study designs. It really needed to be UMI enabled. Ideally, we knew that we wanted to be able to build custom panels from tens of genes to hundreds of genes to address very specific studies and have good sensitivity for those 1% or possibly even below minor allele fraction variants. Really, a future goal is to take this even to deeper coverage and really be able to detect those extremely low allele fractions at less than 0.1% for cases like MRD tracking for patients. So when we built this process or we validated this process, what we were really shooting for was a goal of having at least 90% sensitivity for variants that were at that sort of 1% minor allele fraction. This was just sort of a benchmark for development that we used to build our methods. Also important when you're doing this is we want an extremely low error rate or extremely low false positive rate. And so what we're shooting for is fewer than one false positive per megabase of targeted sequence. Sort of to test our methods once we build them, it's quite simple, the experiments. We actually, to benchmark, we are using 2 healthy donor individuals and spiking one into the other at sort of a decreasing percentage, targeting them with a panel, then sequencing to extreme deep coverage. In this case, we're going for that 20,000x raw coverage. And these are sort of numbers I'm going to start kicking around in this talk because we're thinking about coverage in new ways. We have to sequence extremely deeply. Duplication's not our enemy anymore. It's our friend to help build these UMI consensus reads. So we kind of think about it differently now. The data is deduplicated by duplex UMIs. Calls are generated, and then sensitivity and accuracy are calculated. From this 2 megabase twist pan-cancer panel, in this case, we're looking at sort of like right around a 1% minor allele fraction variant, and we are able to sort of hit that 90% sensitivity when we have a high enough duplex recovery up there, probably over 1,000x coverage. And as you look at the Lego plots for the actual false positives, we really were able to achieve that sort of less than 1 false positive per megabase of sequence. These are just 2 replicate samples done in replicate. So what are our offerings at Broad? We'll take in extracted cell-free DNA, we'll take in blood, and we'll take in pre-fractionated plasma, but we have also automated the system from blood all the way through using the QIAGEN QIAsymphony extraction system. We have a scale of 240 samples per week. That's probably pretty easily scalable. And that's coupled with this Hamilton easy blood imaging-based fractionation. So a really important thing when you're going to spend a lot of money on sequencing deeply a cell-free DNA sample is to hopefully, minimize, but ideally eliminate any sort of genomic contamination of that sample, which completely suppresses your actual coverage of your cell-free DNA that you're interested in. And this really allows that to happen. We always run it in a tumor normal setup where we use the Buffy code as the normal in this case for the analysis. So what we're offering individually or in combination is an upfront ultra low pass whole genome at 0.1x coverage that can give you a tumor purity calculation. And then from there, ideally a lot of things would just go to an exome, and if you have over 10%, say, tumor fraction, 150x is quite useful. But in cases where it's a lot lower, we're going to have to go deeper and probably in custom panels. And again, we're using a duplex UMI analysis and variant calling. So really I want to get into actually the application of Twist target enrichment for what we're now seeing as a pretty dynamic, being able to address real dynamic range of different types of studies. when we saw the performance of our initial effort on the pan-cancer panel, we started to realize how this really fit into a lot of different types of studies. And that's sort of the lesson I want to talk about today. We just saw this slide. We've all seen it a ton of times, and I think for a long time people have seen this and said, at least from the sort of population and disease studies, people say, oh, our N can go up a lot higher. We can do tens of thousands of samples on exome or 20x whole genomes. But really what it ultimately enables also is extremely deep sequencing of fewer samples. And that's sort of what we're getting at, into this new reality of depth. So, for a long time, there's this plot. You want to build a panel. If you want to build a giant panel like an exome, you better be able to run across a lot of samples to justify the upfront development costs and synthesis costs of those probes. Obviously, smaller panels, you still need a lot of samples to actually amortize over. And then there's that little sort of corner down there, which is truly personalized panels, where it might be, 100 probes on a few samples. While it's costly, it's doable. But there's always been sort of this, it didn't have the slide. In the middle there is this sort of like weird nebulous area where we've kind of avoided. We've tried to take a study and sort of shoehorn it into one of these pre-existing designs and just hope for the best with it. But now that we have this third dimension to this plot, which is depth, and now you have target territory, sample number, and now another degree of consideration is how deeply do you want to sequence and how much money do you want to spend on sequencing that depth. A reality that we deal with in the genomics platform, especially in the somatic world, is, in the past 2 years, we've done studies for over 40 cancer types. And, whole genomes, exomes, pan-cancer panels are typically developed. But when you think about it, a lot of you, can you imagine what it takes to get 40 researchers together in a room to decide on what that pan-cancer panel is? And when you try to get it perfect for everybody, what you end up with is probably something that's not ideal for most things. So this is the space that we're thinking about. With Twist, array-based oligosynthesis and the ability to sort of make truly custom panels to your study without a huge upfront cost to test, and without having a huge number of samples to amortize over, we now can envision a world where we now have 40 different panels that are very specific to the disease of interest. If you're only interested in 15 genes in a particular study or clinical study, why sequence another 400 genes to the rest of a pan-cancer panel, especially when you're spending that money to go extremely deeply in a cell-free DNA world? Not to mention, there's also cancer types that aren't necessarily captured or have variation that drives them that aren't captured in an exonic-based targeted panel. So you don't want to leave those out on the side as well. So things like some prostate cancer, osteosarcoma, Ewing sarcoma, there are structural variants that you do want to be able to capture in a very specific study. What we started, this is this dynamic space, and we started realizing that, on the surface of this three-dimensional plot here is, if that's depth, exomes to 20x, germline studies sort of to 20x. But then as you start populating this sort of dynamic range, we realize that we can fill in that space now with the ability to make lots and lots of panels specifically. And this is the reality of where we're going now, is this filling in this whole dynamic range of sample number, target territory, and depth. And while it's technically makes sense now in the lab to do, and I think with the Twist technology and the single workflow that we're building, it actually takes a lot more sort of project management, consultation, custom pricing. So it does kind of open your world up to a little bit of more complexity on that side of things. But technically, it's very valid, and we're excited to start addressing this space the way you see it on the plot here. This isn't just an interesting example of how we were able to take Twist's rapid turnaround, cheap sort of array synthesis, probe synthesis. We had this one study, and some of you may read. It's an interesting article in Wired magazine that's worth a read. Eric Minnickel and Sonia at the Broad, they're very closely interested in studying this prion-driven autosomal dominant disease, fatal familial insomnia. And they just wanted to target the entire PRNP gene, including a bunch of the promoter regions. We were thinking about methods to do it, oligo-based synthesis, or column-based synthesis, and we just realized, let's just send the territory to Twist, have them synthesize, just tile across the whole thing. We're not going to overly worry about repetitive element regions or anything like that. We're just going to sequence it deeply. And in this case, they only have a couple hundred samples in the world to even consider running this against, so you don't want to spend a ton of money up front. And what it did allow us to do is actually map individual variation in the patient population, ultimately with the goal of them designing the actual therapeutics to actually treat this disease. And so that was something that was able to be turned around exceptionally fast without very much overhead, really. This is just another slide where you really start to have to think about like what does depth mean? Like, why are we talking about things to 50,000x coverage? We're excited because what we've built is a sort of a single automated workflow and a single library construction method and Twist panels. And we really believe that we can sort of address this whole range now from germline exome panels where you're only going to sequence to 20, 30, 40x, somatic exomes in that 150x range. But as you start marching down in whether it's cell-free DNA or bone marrow aspirates looking for chip mutations, you're really going to start thinking about sequencing so deeply that you want very specific panels. And that's the reality. To call very low allele fraction variants, we're in the world of sequencing to 50,000x and then deduplicating by those UMIs. So sort of what are the requirements for like a really good working sort of custom panel technology? First off, panel uniformity and evenness, and we've heard that. We've sort of seen sort of the cartoon plots in a couple of the talks. High conversion efficiency, the ability to take a small amount of input material and convert as many of those molecules into sequenceable molecules to get as many unique looks at a variant as possible. A low error rate, so a UMI-enabled library construction method. And if you're going to sequence to 50,000x, you really better hope that most of those reads are on your targets and not distributed all over the genome because you're spending a lot. Rapid turnaround, and then a really good design algorithm, meaning we're able to send a list of targets, get good probe design, that we don't have to go back and do a constant iterative rebalancing or readdressing of that. And we have seen that with like the Twist panels quite a bit. Currently in our pipeline, we now have probably a dozen panels. And in this case, we're ranging from the 35 megabases of a Broad custom panel all the way down to some microsatellite instability panels where we're only talking about 15 kb. What we're really excited about is that single workflow now that we have. Even though we decrease the amount of target territory, we're still seeing those very high % selected rates, and that it just enables us to do a lot of different work. So there's about 12 panels in the pipeline right now that we're working with, and as those come out, I think more and more are going to start filling in. So if we just actually look at some of the performance that we saw, these are some of the first panels, this first 2 megabase pan-cancer panel that we ordered from Twist, and really these are the numbers that drove us deep into sort of the world that we're in now. Fold evenness, fold 80 penalty. These probes we were able to test across 3 or 4 different hybrid selection workflows that we've played with over the years. And no matter really what you do, especially if you're just doing a single capture rather than a double capture, that fold 80 penalty stays pretty flat across the bottom. Fold 80 penalty, for people who aren't familiar with it, is essentially the fold over coverage needed, like how much more sequencing do you need to do to bring 80% of your target bases up to the mean. And that's just the number that sort of we use as an observation of whether it's just assessing the panel performance or even a change in workflow. We want to see that stay pretty consistent. A really nice thing is that's uniform across different panel sizes. You can't really read it terribly well, but on the left is that 2.2 megabase pan-cancer panel, and on the right at only 300 kb. The uniformity is great across the two of them. And if we just look at we've seen the cartoons that Emily showed and others, but if we actually look at IGV coverage of, these are the exact same designs synthesized with two different, an older version of targeting that we used to use, which were biotinylated A-dimers, sort of a first round de novo attempt from Twist to cover regions. We see things like, exons that weren't covered at all being covered. It's extremely even. And MIC, look at this long, it's like this is a 4 kb exon, I think exon 10 and BRCA1, and you can just see the sort of the nice even distribution across the entire exon, in this case of 4 kb. And this is what we're looking for. This is efficient coverage of regions. Again, I mentioned it a little bit before. If you're going to sequence to super deep coverage, you better hope most of those reads are on or near your targets. One of the great things that we saw in early evaluations of the Twist panels is that really no matter what the size is, from large down into small, so 2 megabases down into 300 kb, we still see that high percent selected. This percent selected, in our case, is what comes out of the Picard pipeline, which is percent of bases on or near, so it may vary, there's probably some differences between on target or on bait specific numbers that have been shown earlier. I just want to point that out because it's not that we're necessarily, if it says 80% on bait, that might correlate to our calculation of percent selected from Picard. So now I'd be remiss not to discuss the fact that we are now making the full transition in the Genomics platform to the Twist, a broad custom version of the Twist exome. It's 35 megabases in total, 33 megabases of just the human core exome from Twist, with about 2 megabases of additional content that collaborators, different researchers all kind of chipped in and said this is also things that we really need to have in there. 99% of ClinVar variants are covered. The mitochondrial genome is also spiked in, the full ACMG59 set, and then cosmic variants, and then also the broad ONCA panel genes are also included in the exome. And really, what drives our switch is the plot on the right here, which looks at normalized coverage between our older version of the exome and the pink, which is the newer version of the exome. It's extremely even, and really what that's allowed us to do is hit our deliverable, which is 85% of bases at 20x. That's sort of just how we measure things. Hit our deliverable with a lot less sequencing. Part of that is just really the evenness. Fold 80 penalty is a lot lower. It's allowed for probably about a 50% reduction in sequencing costs for us for germline exomes. I'm specifically talking about germline exomes right now, and soon we will be looking at somatic exomes as well. And hearing the earlier talk about sort of some of the FFP work is really exciting. But what that's also done, this is sort of a little shameful plug, but has allowed us to start offering external exome pricing at $250 now, which you think about, I don't know, maybe 8 or 9 months ago, there was a lot of talk about a lot of that exome work going to 20x genomes. But I think as that price comes down and new technologies pop up like this, it really starts to pull that back to enable the high end for common disease research, big medical population studies still. So all in all, just some observations. So we've built a sensitive and high specificity detection for low fraction variants. It requires high base quality scores, good error suppression, and sometimes in things for low fraction variants, extreme deep coverage. Having flexible and cost-effective panels enabled by Twist's addressable array synthesis really has allowed us to start to explore this dynamic space for these projects. Sequencing is going to get cheaper. We're going to be able to do more samples and go deeper and deeper and deeper to detect those low fraction variants. And what we're proud about is having a single automated workflow, a single library construction set to support things from exome-sized panels down to maybe a couple genes, or even eventually into the MRD space, which would be, individual variants that you would track in a patient over time, post-treatment. So that's where we are. Happy to take questions. Right to 42 seconds left. It's great. Oh, one other thing. So I should acknowledge all the folks in the Genomics platform that have actually done a lot of this work. The folks at Twist have been great to work with. The Broad Cancer Program for spearheading a lot of CellFree DNA work. if people have actually, here's another plug, sort of Broad Genomics services, you can email Genomics at Broad Institute, and we have 2 posters from Jana and Michelle that get into some of the details a little bit more. Thank you.